All right, lecture 3 of photosynthesis. So the reactions, here they are. Hopefully you read this beforehand in your textbook. You're going to hear it now. You're going to copy it down in your notebook, and we definitely will go over this again in class. So as we stated before, photosynthesis is a biochemical pathway that involves a series of oxidation reduction reactions. So it's a redox reaction. And initially what we're going to see is water is going to be oxidized. So when water split, it loses electrons and donates those electrons into this entire biochemical pathway. At the end, carbon dioxide will be the molecule that is to be reduced. There are two basic reactions. There is the light reactions, which is the photo portion of photosynthesis. That occurs in the thylakoid membranes of the chloroplast because the pigments are stored there. And we know that pigments are light-absorbing molecules. So those pigments being stored inside the thylakoids, therefore you have the light reactions. And then the second part is the Calvin cycle, and the Calvin cycle occurs in the stroma because the stroma is where the basically comprises the bulk volume of the chloroplast, and it's where the carbon fixation enzymes are found. And of course, because carbon fixation enzymes are found there, this is where we are going to synthesize our sugar. So what do we know? In the light reactions, which occurs in the thylakoids, we have four main things occurring. One, we're going to have the splitting of water. Therefore, water will become oxidized, therefore donating electrons. And when water is split, we're also going to generate some hydrogen ions, which are going to play a role in the production of ATP via chemiosmosis. We'll also generate some oxygen gas with the splitting of water. We know that oxygen gas is a byproduct of photosynthesis. And at the end there, we will see the formation of NADPH. And NADPH is an electron carrier molecule. And we'll talk about that in class. I will go over that. Then NADPH and that ATP will be used in the Calvin cycle, which occurs in the stroma. And this is the part of the photosynthetic biochemical pathway in which we are going to form our sugar. So the forms of the sugar come from carbon dioxide using ATP, which is our high energy molecule of the cell, and NADPH, which is that electron carrier molecule. So basically NADPH is going to carry electrons from the light reactions to the Calvin cycle. And we could see that here. So if you look at a chloroplast, there you could see the illustration. Here we have the thylakoids. Over here, this would be the stroma area where the Calvin cycle occurs. So in here we have the light reactions, and in the light reactions we get out ATP and NADPH, which gets put in to the Calvin cycle. Coming in, we have water coming in, we have oxygen gas coming out. Over here we have carbon dioxide gas coming into the cycle for the carbon to create our sugar. And coming out of this, we have NADP+, which will then come in back into the light reactions and pick up a hydrogen and electron to form NADPH again. And coming out of the Calvin cycle, we have NADP plus that phosphate group, which then will come back into the light reactions where chemiosmosis occurs and will form more ATP. So, pigments. Basically what happens, in basic chemistry, we looked at electrons, and we said that electrons are what determines the behavior of elements. Well, here you're going to learn that when a pigment absorbs light energy, electrons get excited. So they go from this ground state where they don't have a lot of energy, they're closest to the nucleus of the atom, to an excited state where they're further away from the atom. And when they get excited, they are unstable. Think of you when you're on a complete sugar high. You're all anxious and excited and hyper, but that doesn't last forever because eventually you come upon that sugar crash and then you're tired and you're like, oh. So that's basically what's going on here with the electrons. So when a pigment, such as chlorophyll or the carotenoids, xanthophylls, whatever, when they absorb that light energy, electrons in that chlorophyll are going to get excited. So they're going to go from that ground state to the excited state. And in order to fall back down to that ground state again, they need to release energy. So when excited electrons fall back to the ground state, photons are given off. This would be light energy. It would be an afterglow called fluorescence. And you can see this when you extract chlorophyll out of the thylakoid membranes of a chloroplast. And we did that when we did the pigment chromatography lab. And I'll illustrate this fluorescence process in lab. So fluorescence is when the electrons from chlorophyll come back down and to their ground state from their excited state. And in order to do that, they need to release energy in the form of both light and heat. And here you can see that there. This here is called the cyclic pathway. So the cyclic pathway, basically, if you think of a cycle, what's happening is, here you have your chlorophyll pile. Here are the particles of light energy coming in as photons, and electrons go from a ground state up to an excited state, and as they fall back down, they release heat and light energy, and that would be fluorescence, and then they come back down. So it's like one complete cycle because the electrons are no longer stored in the thylakoid compartments. So the pigments there just keep on cycling those electrons from an excited state to a ground state, and that would cause the extracted pigment to fluoresce. Now in photosynthesis, it's not a cyclic pathway, it's a non-cyclic pathway. And when we look at photosynthesis and its non-cyclic pathway, we are going to be using the discussion of photosystems. And a photosystem consists of a reaction center surrounded by light harvesting complexes. These light harvesting complexes are pigment molecules that are bound to proteins. And what they do is they're going to funnel the energy of photons, which is light, to this reaction center to excite the electrons. So the primary electron acceptor in the reaction center accepts an excited electron from chlorophyll A. And then this solar-powered transfer of an electron from chlorophyll A molecule to the primary electron acceptor is the first step to the light reactions. And I have a detailed diagram illustrating all of this. So if we look here, we could see that reaction center. So here we are, here's the stroma. So we're outside the thylakoid compartment. Here's that thylakoid membrane that I told you about. And like all biological membranes, you can see the phospholipids. Here is that special reaction center. And then down here we are inside the thylakoid space, so inside the thylakoid compartment. So here we have light energy coming in. Here we see these pigment molecules, these chlorophyll A molecules, and they're going from a ground state up to this reaction center excited state because up here we have those light harvesting complexes. So this here, this little box here would represent the primary electron acceptor. Now, there are two types of photosystems. There's photosystem 2, which functions first. The numbers reflect the order of discovery. So basically, photosystem 1 came about first, and photosystem 1 is best at absorbing wavelengths of light at 700 nanometers. And then later we discovered photosystem 2, and actually photosystem 2 is where this all begins. It functions first, and it's the best at absorbing a wavelength of light energy at 680 nanometers. So the two photosystems work together to use light energy to generate ATP and NADPH. So in the non-cyclic electron flow during the light reactions, there are two possible routes for electrons, the cyclic or non-cyclic. We're going to look at this non-cyclic electron flow in which the primary pathway involves both photosystems to produce that ATP and NADPH molecule. And if we look here, you can see it. So this is a basic diagram of where we have water coming in. Water is going to split to generate oxygen gas. We're also going to generate hydrogen ions. Up the left side of the diagram here, you can see the energy of electrons. So here at photosystem 2, light energy is best absorbed at 680 nanometers, and when that light energy comes in, those electrons get excited and go to their excited state. And there you have that primary electron acceptor. That would be plastoquinone. So then as those electrons travel through the thylakoid membrane, through those cytochrome complexes, they're going to pass through, and as they come back down to their ground state, which would be in photosystem 1, energy needs to be released. And that energy released is going to be used to make ATP. This is called the electron transport chain, and the electron transport chain is coupled with chemiosmosis in ATP production. Now, over in photosystem 1, basically what's happening, again, you have electrons now in their ground state. Light energy is being absorbed by the pigment harnessing complexes there, and it's best absorbed at 700 nanometers of light. Those electrons are going to go from a ground state back up to an excited state to the primary acceptor of electrons, and then as they pass down, they're not going to go back down into a third photosystem. What happens here is an enzyme is going to be used, and here you can see NADP plus reductase. An enzyme is going to be used to reduce NADP plus, pick up 2 hydrogen ions, and you're going to form NADPH. NADPH plus and H plus. So NADPH is an electron rich, and that's why the enzyme's name is given NADP plus reductase. It's a reduction reaction here. So NADPH is going to be reduced to form NADPH, and in doing so, you also have 2 hydrogen ions coming in. One is going to become part of that molecule. The other one is kind of left there. So that is our electron carrier molecule. And basically, ATP and the generated NADPH is going to be used in the Calvin cycle in the stroma. The oxygen gas that was generated, that will be expelled through the stomates when they are open back into the atmosphere. Now, that is the non-cyclic electron flow. The cyclic electron flow, in a cyclic electron flow, it uses only photosystem 1, and it produces only ATP. So the cyclic electron flow generates a surplus of ATP, satisfying the higher demand in the Calvin cycle. So here you can see that here. In photosystem 1, you keep rotating back electrons, and it comes back in, and you keep getting that production of the ATP there. So what is chemiosmosis? Chemiosmosis occurs in both photosynthesis cell respiration, so the chloroplast and mitochondria, and it's a process used to generate ATP, and but it uses different sources of energy. In the mitochondria transfer chemical energy from food to ATP. In the chloroplast, it transforms light energy into the chemical energy of ATP. So the spatial organization of chemiosmosis differs in both the chloroplast and the mitochondria. So here you can see basically what's happening in the electron transport chain and chemiosmosis. But to get a better understanding of what's going on in photosynthesis, we need to connect what's happening in the beginning of photosystem 2. So in photosystem 2, water is split, and then on the side of the membrane facing the thylakoid space, and when water is split, you get the diffusion of hydrogen ions from the thylakoid space back to the stroma, which is going to be used to power ATP synthase, which is an enzyme used to make ATP. And then you have ATP and NADPH that are going to be produced on the side facing the stroma, where the Calvin cycle takes place, because you want that production of those molecules to be there, because they are both going to be used in the carbon fixation reaction, or the Calvin cycle. So if we put this whole grand scheme together, and here's a basic diagram of what's going on, here you can see photosystem 2 passing photosystem 1. So water split, generate an oxygen gas, you generate hydrogen ions. So inside the thylakoid compartment, you generate a high concentration of hydrogen ions, which makes it more acidic. And then those electrons will pass through from an excited state to the ground state through the thylakoid membrane, from photosystem 2 to photosystem 1, where they then get re-excited again, and you form NADPH. This would be the ETS system. As the electrons pass through the thylakoid membranes, the energy extracted is going to be used to pump hydrogen ions against the concentration gradient from the stroma, where they're in low concentration, into the thylakoid compartment, where they're in high concentration. Now we'll have facilitated diffusion occurring, chemiosmosis, where they pass through this channel protein known as ATP synthase from an area of high concentration into an area of low concentration to make ATP. That's chemiosmosis.